Okay, so this is lesson 1 of haemoglobin. What we've got here is a graphic of haemoglobin. And so let's get into what haemoglobin is. Hopefully you've all heard of haemoglobin from GCSE and know that when it's combined with oxygen, it's called oxyhemoglobin. Haemoglobin is the molecule, a very large molecule, but it's a molecule that carries oxygen. So, a little bit of a recap on proteins. Remember, as you move through the course, you'll be expected to use all the knowledge from you that you've already gone through. So here we need to know about protein structure, amino acid sequencing, transcription and translation, mutations, etc. Okay, so what are the monomers that make up proteins? This should be an easy peasy question. They are amino acids joined with a peptide bond. What is the primary protein structure and what bonds holds it in place? So the primary protein structure is the sequence of amino acids or the order of amino acids within one polypeptide chain, and they're connected with peptide bonds. And you should be able to draw a dipeptide and draw what the peptide bond looks like. It's the carbon of the carboxyl group is bound to the nitrogen of the amine group of the adjacent amino acid. What is the secondary protein structure? So it's where the individual polypeptide is coiled or folded into sheets, and these are held in place with hydrogen bonds. What is the tertiary protein structure? So it's how the polypeptide chain is then folded in on itself, and these bonds that holds that shape is disulfide bridges, ionic bonds, and then lots more hydrogen bonds as well. It depends what amino acid you've got in that polypeptide chain and in what position they are, what position those possible ionic or ionic bonds and disulfide bridges are, which allows it to have a certain shape. Obviously, if you have different amino acids, the bonds are going to be in different places, so you're not going to have the same shape. So that's how the DNA sequence links to the primary structure, which then links to the tertiary structure. And the tertiary structure can ultimately determine its function, whether we know that enzymes and carrier proteins, haemoglobin, are very dependent upon their shape. What is the quaternary protein structure? So this is if it's got more than one polypeptide in it, and possibly non-protein groups that are associated with it. So haemoglobin is a protein and it's made up from 4 polypeptide chains. Each one is associated with a heme group. This diagram here, this little pink structure, this is a heme group. It's a very large organic molecule with a ring structure and then inorganic iron in the middle of it. Each heme group carries one oxygen molecule. So one oxygen molecule binds to the iron in the heme group. Okay, and when it's associated with oxygen, it's called oxyhemoglobin. Oxygen can bind to haemoglobin reversibly, so we want that. We want it to bind to haemoglobin in the lungs, and then we want it to unbind or dissociate. Sometimes we might say unload the oxygen in the respiring tissues where it can be used for respiration, aerobic respiration. Each living cell requires oxygen for aerobic respiration to release the energy or to produce ATP required for functions of the cell. True or false? Hemoglobin is made up of 3 polypeptide chains. False, it's 4. Hemoglobin can carry 4 oxygen molecules. True, yes it can. When oxygen joins to haemoglobin, it forms oxyhemoglobin. True. And the shape of haemoglobin is determined by its secondary structure. Well, it's more its primary structure. Okay, so how is it adapted for its function? So this is a little diagram. This is supposed to be a capillary here. This sort of disc is supposed to be a red blood cell, and these are all the little haemoglobin molecules inside. Remember, red blood cells don't have a nucleus, so they've got more room to carry haemoglobin. They're like just little packages of haemoglobin, basically. As the blood moves through the capillaries across the alveolar surface, oxygen diffuses from a high concentration to a lower concentration through the epithelium of the alveoli and endothelium of the capillary into the red blood cells, where it will bind with a heme group to form oxyhemoglobin, and then it will then leave the alveolar surface, and blood which is low in oxygen will replace it. Then it will go back to the heart. The heart will then pump it, goes in the left side, and the heart will then pump it out through the aorta to everywhere around the body. So oxygen or haemoglobin readily binds with oxygen at gas exchange surfaces where there is a high partial pressure of oxygen, or where there's a high concentration of oxygen, but we use the term partial pressure when we're talking about gases. So where there's a high partial pressure of oxygen, haemoglobin will bind readily to it. Then the circulatory system pumps the blood to the tissues, where this is an example, some muscles. We've got some respiring cells where you want oxygen to unbind now, and then diffuse from the haemoglobin into the tissue fluid, from the tissue fluid into the cells, and from the cells into the mitochondria, where it be used in aerobic respiration. So it readily unloads oxygen in tissues where there is a high concentration of carbon dioxide, so lots of carbon dioxide is being produced through aerobic respiration. This almost acts as a trigger to allow the haemoglobin to unload the oxygen into the respiring tissues. So where there's a high carbon dioxide concentration and a low partial pressure of oxygen, the haemoglobin will unbind to the oxygen or unload the oxygen. Haemoglobin therefore has the ability to both load readily and dissociate easily, and both of these are important. You need it to actually bind to the oxygen, but you need it to let go of it when oxygen is required. It does this because the haemoglobin can change shape because of the presence of carbon dioxide. Also, you actually get different types of haemoglobin. So remember we said that the shape, the tertiary structure is dependent upon the primary structure. The primary structure is dependent upon the DNA sequences. All organisms will have slightly different DNA sequences for haemoglobin in the haemoglobin gene. Well, there'll be a gene for the two different types of polypeptides. So there might be a slightly different order of nucleotides in the gene for haemoglobin depending on the species, and that has that particular frequency of that allele will have increased due to natural selection. So depending on the habitat, size and activity of the organism, that's going to affect their oxygen demand and therefore affect the shape of the haemoglobin molecule. So just a few little ideas here to get you thinking. So this is a mountain goat. It lives in areas with a very high altitude. What do we know about high altitude? We know that as you move up, actually the concentration within the of oxygen in the air decreases. You say the air is thin. There's a slightly lower concentration of oxygen in air the higher you get. So for the mountain goat, it needs to be able to extract oxygen from the air a lot more easily when the partial pressure is lower. So we need here for the mountain goat a haemoglobin that has a very high affinity. Affinity is the word we use to describe the sort of attraction between oxygen and haemoglobin, the likelihood that it will bind. Okay, so if haemoglobin has a high affinity for oxygen, it means it binds it more readily, more easily at lower partial pressures. So there's less oxygen available, so it needs to load oxygen more easily. Their haemoglobin has a higher affinity for oxygen, so it can become saturated at a lower partial pressure. Okay, so where oxygen partial pressure is lower, it will load oxygen more easily than, say, our haemoglobin. Now there's a few more here, so we've got the answers here, so I'll just discuss them. So these three different organisms here. Here we have a little mouse. What do we know about mice? We know that they're small and they're mammals, so they're warm-blooded. Warm-blooded animals give off heat. This mouse is small. Heat loss, small, what do we know? Small organisms have a higher surface area to volume ratio, so they lose heat more quickly. So, what do organisms do in order to maintain their body temperature? They respire, because heat is a waste product of respiration. So, what do we need for respiration? We need oxygen. So this little mouse is going to need to have lots of oxygen unloaded at its tissue so it can respire lots, give off lots of heat to replace the heat that's being lost due to its large surface area to volume ratio. Okay, this is a eagle. It has, it does fly higher, but it can come down to other altitudes. But flying is very energy intensive. Okay, so it has a very high demand for oxygen, so it's a very high metabolic rate. So therefore, it requires lots of oxygen to be unloaded in the tissues, to be able to carry out respiration, to be able to keep contracting those muscles required for flight. Okay, so this eagle needs haemoglobin with a low affinity. This mouse needs haemoglobin with a low affinity. If it's got a low affinity, the haemoglobin is going to give up its oxygen really easily. If haemoglobin's got a high affinity, it's going to hold on to that oxygen. Okay, so it will give up its oxygen at higher partial pressures. And the eagle its haemoglobin will give up its oxygen at higher partial pressures. Our haemoglobin, you would need it to get to a much lower oxygen concentration, partial pressure, for it to actually unload the oxygen, whereas their haemoglobin is slightly different and it unloads its oxygen at slightly higher partial pressures of oxygen. And then the final one is a fetus. So where does a fetus get its oxygen from? From the mother's blood. Okay, so the mother's blood contains haemoglobin, and the haemoglobin is associated and bound to the oxygen. What we need in the fetus is we need a special type of haemoglobin in the fetus that's got a higher affinity for oxygen than the mother's haemoglobin. And if it's got a higher affinity, it's basically going to snatch that oxygen from the mother's haemoglobin. If they had the same affinity, the mother's haemoglobin would not give up that oxygen. But because the fetal haemoglobin has a higher affinity, it will actually almost steal the oxygen from the mother's haemoglobin. It allows it to be unloaded much more easily at the placenta. Okay. When the baby is born, due to gene expression, they'll stop expressing the genes to make fetal haemoglobin, and they'll start expressing the gene to make normal haemoglobin once they've been born and they're not using the placenta anymore. Next lesson, we're going to look, later on in this lesson, you're going to look at the oxygen dissociation curve. And next lesson, we're going to look at variants of that curve and what can affect that curve.